Welcome to KMR Advice. We're excited to share helpful tips and advice with you. Check out our latest presentation, Mechanisms of Drug Transport Across Cell Membranes, a comprehensive overview. Subscribe and join us on this journey. Pharmacokinetics explain what the body does to drug, and pharmacodynamics explain what the drug does to body. These processes help us understand the effects and interactions of drugs within biological systems. Absorption, distribution, metabolism, and excretion are key components of pharmacokinetics. They determine how a drug is processed and ultimately eliminated from the body. Drug administration begins with routes like IV or oral, leading to absorption into the plasma. In plasma, drugs exist as bound to proteins or free and active, allowing free drugs to be distributed to tissues and sites of action. Some drugs are stored in tissues, affecting their duration of action. Metabolism occurs primarily in the liver, transforming drugs into metabolites. Finally, drugs removes by excretion through kidneys, bile, or exhalation, completing the journey of a drug in the body from administration to excretion. Drugs move transmembranely through extracellular and intracellular pathways, utilizing water-filled pores and carriers. This movement is essential for reaching target sites. Drug passage across cell membranes occurs via two main processes, first passive transfer and second specialized transport. Passive transfer, its two types. First, simple diffusion, moves drugs from high to low concentration without energy. Second, filtration, uses pressure differences to pass drugs through membrane pores. Specialized transport, its three types. First, facilitated diffusion, utilizes carrier proteins, no energy needed. Second, active transport, moves drugs against the gradient using energy. Third, pinocytosis, engulfs drugs in vesicles for transport. Let's focus on simple diffusion, a key component of passive transfer. Simple diffusion is the most important process for drug movement across membranes. Here's why. Drugs move along the concentration gradient, flowing naturally from areas of high concentration to low. This process requires no energy, making it efficient. No carrier proteins are needed, allowing drugs to pass freely. Simple diffusion is not saturable, meaning it can continue without reaching a maximum capacity. Additionally, it is not inhibited by metabolic inhibitors, ensuring consistent drug passage. Filtration, another essential mechanism of passive transfer. Filtration involves the movement of drugs through water-filled pores in the cell membrane. Here's what makes it unique: it is not affected by lipid solubility, allowing even water-soluble drugs to pass through. Filtration is not saturable, meaning there is no limit to the amount of drug that can be filtered through these pores. Continuing with filtration, let's explore how this process is influenced by certain pressures. Filtration through capillaries depends on hydrostatic pressure. This is the force exerted by the fluid against the capillary walls, driving the movement of drugs through the pores. Osmotic pressure, this pressure results from the solute concentration, affecting the movement of water and solutes across the membrane. Blood flow, filtration is limited by blood flow, meaning the rate of drug passage can be affected by changes in circulation. Facilitated diffusion, This process is similar to simple diffusion but requires specific conditions. Facilitated diffusion requires a carrier, meaning it can reach a maximum rate of transport when all carriers are occupied. This mechanism ensures that essential molecules can efficiently enter or exit the cell, even if they can't diffuse directly through the lipid bilayer. Continuing with specialized transport, we now explore active transport. Active transport is a process that moves substances against their concentration gradient. This means moving particles from an area of lower concentration to an area of higher concentration. This process requires energy, typically in the form of ATP, to power the transport. An example of active transport is the renal tubular secretion of organic acids, which is crucial for maintaining proper balance and function in the kidneys. Active transport ensures that cells have the necessary molecules even when their concentration is higher inside the cell than outside. Next, we explore panocytosis, an energy-dependent process. Panocytosis involves the engulfing of large molecules inside cells. This mechanism is crucial for absorbing substances such as vitamin B12 and intrinsic factor. During panocytosis, cells actively take in extracellular fluid and its contents, allowing them to acquire nutrients and other essential molecules. This process highlights the dynamic nature of cellular transport, ensuring cells receive vital components for their function. Thank you for joining us on this journey. Don't forget to like, share, subscribe, and hit the notification bell for more insightful content. If you have any questions or thoughts, feel free to leave them in the comments below.